Welcome! In this video, we are going to cover an introduction to the Baltimore classification. This will evidently be very distinct from the ICTV by examining how a virus may replicate itself, make its messenger RNA, and make proteins. For the purpose of this course, we will only be covering 6 of the 7 groups, ignoring the double-stranded DNA pararetroviruses. Notice how all of these classes share the common theme of mRNA synthesis, highlighted in yellow in the middle. We will be utilizing this theme in the accompanying worksheet to practice viral replication. What is a virion? A virion is an infectious particle that is transmitted from one host cell to another. The minimal constraint is a genome protected by capsid proteins. The genome, in the bulletin point, is written as nucleic acid because this may be either RNA or DNA. In this top image, we can see the gray triangular capsid proteins forming one complete capsid coat. In this little window pane that I have opened up, we can see the genome in red. Specific viruses may also have an envelope derived from the host membrane. The phospholipids of this envelope are always from the host, so this gray border would be from the host membrane. However, the virus may direct the production of certain envelope proteins. So the black spikes here, or spike proteins, could be made by the viral information. For this exercise, we will need to consider the machinery that is used to reproduce a complete virion. Every virion must contain a complete copy of the genome, so we will need to discuss how to replicate, transcribe, and translate the genome. A good portion of the genome types will have special machinery that must be used, and we will then question how that machinery is brought along to future virions. Packaged versus encoded. Encoding is a set of instructions provided in the genome. Encoded proteins are required by the virus to make complete functional virions. These proteins may undergo production in the cell if the proper machinery is present to transcribe and translate. Packaging indicates that the protein is included in the virion. It must be brought along for reproduction to occur. Packaging and encoding are not mutually exclusive. If a protein is packaged, it must be encoded. This ensures that all progeny can receive a copy of the protein and can go along to be functional in additional hosts. Capsid proteins, for example, are always encoded by the virus. The virus needs the capsid for protection, as well as attachment and entry in certain cases. Oddly enough, we consider this one to be packaged by the virus too. It's a little trippy because this protein may act as the exterior-most coat. So don't get confused if we call it packaged, but it is that outermost layer. Now, take a moment to consider, why is encoding not enough? Why do we ever need to package a protein? Have you ever been disappointed by a toy that came with no batteries? The toy is non-functional. The toy may have included a detailed instruction pamphlet, but without any batteries, the toy will never work. Now for the virus, it could provide a full set of encoded instructions, but without the extra packaged components, it cannot successfully propagate itself. Viruses are acellular and must infect a host cell to reproduce. Before we get into the nitty-gritty, we need to understand what machinery is available for hijacking in the typical cellular host. So, as this image displays on the right, we go from DNA. We can produce mRNA through the assistance of an RNA polymerase. And from the mRNA, we use ribosomes to create a polypeptide and eventually proteins. So, the central dogma describes the flow of information all the way from DNA to proteins. Hopefully you're familiar with this molecular process of replication, transcription, and translation. An introductory biology course would mention the key players of each of these steps: DNA polymerase, RNA polymerase, and ribosomes. A polymerase is an enzyme that assists in the synthesis of nucleic acids like RNA or DNA. The resulting strand is complementary to the template strand. A's to T's, G's to C's for DNA, or A's to U's and G's to C's for RNA. Polymerases come in many different forms. We have a specific nomenclature for polymerases: template-dependent product polymerase. Consider what polymerases could be used for molecular processes in a cellular organism. Replication begins with DNA and ends with DNA, so according to our nomenclature, it should be called a DNA-dependent DNA polymerase. Transcription begins with DNA and ends with RNA, so according to our nomenclature, it should be called a DNA-dependent RNA polymerase. The use of a template for polymerase action results in an anti-parallel complementary strand. This can be seen in the example figure. The backbones in purple of the nucleic acid are in opposite directions. Their directionality is determined by the phosphate end and the hydroxyl end. Meanwhile, these bases complement each other, giving it the full name of anti-parallel and complementary. According to the previous slide, the opposite directionality comes from the difference in reading versus synthesizing. Polynucleotide synthesis takes place in the 5' to 3' direction, whereas the template is read in the 3' to 5' direction. The Baltimore virus classification system is based on genome composition and means of mRNA production. Complete the accompanying worksheet to practice the replication and transcription processes undergone by the virus. The first piece of instruction gives you an example strand of the mRNA. Hopefully, it can become clear that each class has a unique genome, but shares the common theme of mRNA as we introduced earlier in this video. Using this information, let's see how we're going to synthesize the genome and the messenger RNA. In the following cases, we will be referring to the viruses by their genome. We'll start with perhaps the most intuitive genome, double-stranded DNA. Attempt this portion of the worksheet before proceeding. Pause the video if necessary. The mRNA is displayed in the top corner of this video whenever we introduce a new class of virus. If we are working backward to find the genome, then the polymerase must have read the strand from 3' to 5' in order to have synthesized this mRNA strand. Hence, we can deduce a 3' to 5' antiparallel and complementary strand to the mRNA. If we tried practicing this, we would write a 3' strand, and it would be written T, T, C, T, A, G, T, G, A, and then a 5' end. Meanwhile, the 5' to 3' strand of the DNA must have looked pretty identical to the mRNA, but using T's instead of U's. A complementary and antiparallel strand to the mRNA can be called the negative strand. The strand resembling the mRNA can be called the positive strand. As we've discussed in this course, double-stranded DNA is the genetic information to all cellular life. Thus, for the host genome to undergo the essential processes of transcription and translation, the host must already have some machinery available for the virus to utilize. DNA-dependent DNA polymerase is found within the host cells. It will replicate the double-stranded DNA virus. Meanwhile, DNA-dependent RNA polymerase is also found within the host and will transcribe the viral double-stranded DNA to mRNA. Ribosomes will always perform the translation and are found within the host. For the purpose of this course, we will assume that the host cell provides ddDP and ddRP. This is not always the case. Viruses may encode and thereby provide ddDP and/or ddRP. As we've discussed in this course, double-stranded DNA is the genetic information to all cellular life. Thus, for the host genome to undergo the essential processes of transcription and translation, the host must already have some machinery available for the virus to utilize. DNA-dependent DNA polymerase is found within the host cells. It will replicate the double-stranded DNA virus. Meanwhile, DNA-dependent RNA polymerase is also found within the host and will transcribe the viral double-stranded DNA to mRNA. Ribosomes will always perform the translation and are found within the host. Positive single-stranded DNA. The positive single-stranded DNA genome will look identical to the mRNA. However, it must have thymines instead of uracils. Pay attention to the direction on single-stranded molecules. You just might hit a case where you need to answer from the opposite directionality, 3' to 5'. Positive single-stranded DNA machinery requirements. Cellular life will have the DNA-dependent DNA polymerase needed to replicate and the DNA-dependent RNA polymerase required to generate mRNA. To keep things consistent, the replication cycle is drawn on the top path and the transcription translation is on the bottom path. Replication might look unfamiliar because this is rolling circle replication. First, the single-stranded DNA is made double-stranded, and then another single-stranded molecule of DNA can be produced. Meanwhile, the transcription translation path should look pretty familiar. However, we can't simply use the single-stranded DNA alone to make mRNA. We have this intermediate of a double-stranded DNA molecule. So DDDP will make a double-stranded molecule, and then DDRP will take that double-stranded molecule and make mRNA, eventually processed in ribosomes to make proteins. Now we are entering the RNA portion of these genomes. For positive single-stranded RNA, the genome is actually identical to the mRNA. Onto the machinery requirements. We consider that positive single-stranded RNA can be used as mRNA. And because mRNA is found in cellular life as the temporary transcript between double-stranded DNA and proteins, host cells already contain the proper machinery to make proteins for this type of viral genome. However, the cell lacks the ability to replicate the RNA. No cellular machinery can synthesize RNA from an RNA template. For this process, the virus must encode RNA-dependent RNA polymerase. In the top half of the diagram, positive single-stranded RNA will act as a template in RDRP, generating a negative single-stranded RNA molecule. Putting this into a virion would result in the wrong code, anti-parallel and complementary, being propagated in future hosts. Hence, one more cycle of RDRP replication must be performed to synthesize positive single-stranded RNA. In the bottom path of the diagram, you'll notice the positive single-stranded RNA can act directly as mRNA and use the ribosomes to make proteins. Those proteins include the encoded RDRP. So, because RDRP can be made unhindered from additional machinery requirements, we do not need to package the physical copy of RDRP. Negative single-stranded RNA. Negative single-stranded RNA is the anti-parallel complementary strand to the mRNA. Now be extra careful about the directionality for this set, because you may be given a question that asks from 5' to 3', or you may be given an instance where you need to write it from 3' to 5'. So, as this little animation shows, you would need to flip-flop where the 3' end is and where the 5' end is, and you would simply take each nucleotide and write them in the reverse order. Now for the machinery requirements. Unlike the positive single-stranded RNA, RDRP must process the RNA for replication. However, in this case, the RDRP is also required for mRNA before proteins can be made. Both paths need RDRP and no proteins can be formed without this polymerase, so it must be packaged. RDRP must also be encoded to ensure that all virions also have a copy of the polymerase for their future propagation into additional hosts. Otherwise, the virus would be stranded when it reaches the next host. Note that the proteins could theoretically be made in the negative direction, however, these would be utter garbage. The desired proteins are coded in the positive orientation. So you'll notice in this diagram, we have the negative single-stranded RNA in yellow being processed by RDRP in order to make a positive single-stranded RNA, and that needs to be processed once more to get back to the original genome. And in the other direction, we have the negative single-stranded RNA being processed by RDRP in order to make mRNA, and that can be used by ribosomes to make proteins. Noticing that the RDRP is required in both paths, so it must be packaged and encoded, otherwise the virus is stuck with nothing to do. Double-stranded RNA. Based on each virus, only one of the strands of double-stranded RNA is used as the template for mRNA. Just as in cellular organisms, generally only one strand of the DNA codes for the mRNA and the corresponding proteins. Keep in mind that if we have double-stranded RNA, we will not be seeing any T's in the replication process. So, one of our strands is identical to the mRNA, and one is complementary and antiparallel. No T's. Double-stranded RNA machinery requirements. With a double-stranded molecule, we have to question how the molecule is going to break open in order to allow the machinery to process one of the strands. Since cells do not have an RNA helicase, the virion must not only encode but also package RDRP, which has helicase activity. Replication is going to use this RDRP, and proteins cannot be made without the RDRP. So we must package and encode RDRP in order to have a functional double-stranded RNA virus. In the top path, RDRP helps with replication. Bottom path, RDRP helps in the mRNA production for ribosome processing. Retroviruses. Retroviral genomes are identical to the mRNA, as it is a positive single-stranded RNA genome. However, this group of viruses undergoes an additional step of an intermediate double-stranded DNA formation, which will integrate into the host chromosome for viral reproduction. And this viral genome survives within the host. So, we have our molecule looking identical to the mRNA. Onto the machinery requirements. Retroviruses have an RNA genome that will be inserted as double-stranded DNA into the host chromosome. Clearly, this isn't a function that the host normally performs, so we're going to need to call on some special machinery. First step is reverse transcriptase. This will use a positive single-stranded RNA molecule to make DNA. There are three functions of reverse transcriptase: RNA-dependent DNA polymerase, making that first strand of the double-stranded DNA, nuclease, that will remove the single-stranded RNA from that hybrid molecule, and DNA-dependent DNA polymerase, which will make the second strand of the double-stranded DNA. Second, in the chain of operations, integrase will insert the viral content into the host chromosome. Here, the host chromosome is shown in yellow, and the viral genome is shown in green and blue. Not shown, the entire genome, host with the integrated viral genome, is replicated by host DNA-dependent DNA polymerase. Following, the viral genome is transcribed by host DNA-dependent RNA polymerase when ready. Viral mRNA is then sent through ribosomes, forming a long viral protein. In certain cases, proteases are required to cleave the long product into functional units like additional reverse transcriptase molecules, integrases, proteases, capsid proteins, spike proteins, and more. As written in the slide, it is important that these proteins are both packaged and encoded for complete propagation of the virus. This case concludes the classes that are covered for this course.